Welcome to this exploration of cancer, one of the most significant diseases affecting humanity. Cancer is fundamentally a disease of uncontrolled cell division. In this video, we will understand how failures in the cell cycle lead to cancer development. This topic is crucial because it connects what we have learned about normal cell division to real-world medical conditions. By understanding cancer at the cellular level, we gain insights into why it develops and how it can be treated. So what exactly is cancer? Cancer is a disease where cells in the body begin to divide uncontrollably and spread to surrounding tissues. Unlike normal cells that follow strict rules, cancer cells ignore the body's signals to stop dividing. The root cause of cancer lies in the breakdown of normal cell cycle regulation mechanisms. Think of it like a car whose brakes have failed. The engine keeps running, but there is no way to stop it. In cancer, the cellular machinery that controls division is damaged, leading to endless, uncontrolled growth. Let us compare normal cells with cancer cells to understand the key differences. Normal cells exhibit controlled growth, stopping when they receive the appropriate signals from the body. They remain in their designated location and even self-destruct if they become damaged, a process called apoptosis. Cancer cells, on the other hand, grow uncontrollably and completely ignore stop signals from the body. They invade nearby tissues, spreading beyond their original location. Most dangerously, they evade programmed cell death, continuing to multiply even when damaged. These characteristics make cancer cells fundamentally different from healthy cells. Cancer arises from three main causes. First are genetic mutations, which are changes in the DNA that affect genes controlling the cell cycle. These mutations can occur randomly or be triggered by external factors. Second are environmental factors, including exposure to radiation, harmful chemicals, certain viruses, and lifestyle choices like smoking or excessive sun exposure. Third are inherited genes. Some people inherit mutations from their parents that increase their cancer risk. However, it is important to note that inheriting a cancer gene does not guarantee cancer development. It simply means a higher susceptibility. Most cancers result from a combination of these factors working together over time. Two major types of genes play critical roles in cancer development. The first type is proto-oncogenes. These genes normally promote healthy cell growth and division. However, when they become mutated, they transform into oncogenes that drive excessive, uncontrolled cell division. Think of them as the accelerator pedal in a car that gets stuck down. The second type is tumor suppressor genes. These genes act as the brakes of the cell cycle, slowing down division and checking for errors. When tumor suppressor genes lose their function through mutation, the cell loses its ability to control growth. Without functioning brakes, cell division proceeds unchecked, leading to tumor formation. Let us examine a specific example, the p53 gene, often called the guardian of the genome. In its normal role, p53 is crucial for preventing cancer. When DNA damage is detected, p53 stops the cell cycle, allowing time for repair. If the damage is too severe, p53 triggers cell death to prevent the damaged cell from dividing. However, when the p53 gene itself becomes mutated, these protective functions are lost. Damaged cells continue dividing, accumulating even more mutations with each division. The impact of p53 mutations is enormous. This gene is found mutated in over 50% of all human cancers, making it one of the most important genes in cancer biology. Cancer develops through three distinct stages. The first stage is initiation, where a single cell acquires its first mutation. This mutation might damage a gene that controls cell division. At this point, the cell is not yet cancerous. The second stage is promotion. During this phase, the mutated cell begins to multiply, forming a mass of cells called a tumor. Additional mutations may occur during this stage, making the cells more abnormal. The third and final stage is progression. This is when cancer cells become invasive and spread to other parts of the body through the bloodstream or lymphatic system. This spreading process makes cancer much more difficult to treat and is responsible for most cancer-related deaths. Metastasis is the process by which cancer spreads from its original location to distant parts of the body. This is what makes cancer particularly dangerous. Metastasis occurs in 4 key steps. First, cancer cells detach from the primary tumor, breaking the connections that normally hold cells together. Second, these cells invade into nearby blood vessels or lymphatic vessels, gaining access to the body's circulatory systems. Third, cancer cells travel through the bloodstream or lymph system to distant organs. Finally, these traveling cells exit the vessels and establish new tumors in other locations, such as the lungs, liver, bones, or brain. Metastatic cancer is more challenging to treat because it affects multiple body systems simultaneously. Modern medicine employs several approaches to treat cancer. Surgery involves the physical removal of tumor tissue and is often the first line of treatment for solid tumors. Radiation therapy uses high-energy rays, such as X-rays, to kill cancer cells by damaging their DNA. Chemotherapy employs powerful drugs that target rapidly dividing cells throughout the body. While effective, chemotherapy also affects healthy, fast-growing cells, causing side effects. Targeted therapy represents a newer approach, using drugs designed to attack specific molecules found in cancer cells. This precision approach often has fewer side effects than traditional chemotherapy. Most cancer patients receive a combination of these treatments tailored to their specific cancer type and stage. Let us review the key concepts from this video. First, cancer fundamentally results from failures in cell cycle regulation mechanisms. When the checkpoints and controls that normally govern cell division break down, uncontrolled growth occurs. Second, mutations in 2 types of genes drive cancer development: proto-oncogenes, which become overactive, and tumor suppressor genes, which lose their protective functions. Both types of mutations disrupt the careful balance that keeps cell division under control. Finally, understanding how cell division works and how it fails in cancer has enabled scientists to develop increasingly targeted and effective cancer treatments. This knowledge continues to drive medical advances, offering hope for better treatments and ultimately cures for various types of cancer.
If you like this video, hit that like button and don't forget to subscribe. Visit CodeLucky.com for more such useful content.